Breakthrough: brain cells protect muscles from wasting away. I'm Steve Belair. Welcome to Breakthroughs. While many of us worry about proteins aggregating in our brains as we age and potentially causing Alzheimer's disease and other types of neurodegeneration, we may not realize that some of the same proteins are aggregating in our muscles, setting us up for muscle atrophy at old age. University of California, Berkeley scientists have now found brain cells that help clean up these tangles and prolong life, at least in worms and possibly mice. This could lead to drugs that improve muscle health or extend a healthy human lifespan. The research team's most recent discovery, published January 24th in the journal Science, is that a mere 4 glial cells in the worm's brain control the stress response in cells throughout its body and increase the worm's lifespan by 75%. That was a surprise, since glial cells are often dismissed as mere support cells for the neurons that do the brain's real work, like learning and memory. This finding follows a 2013 study in which the UC Berkeley group reported that neurons help regulate the stress response in peripheral cells, though in a different way than glial cells, and lengthen a worm's life by about 25%. In mice, boosting neuronal regulation increases lifespan by about 10%. Together, these results paint a picture of the brain's two-pronged approach to keeping the body's cells healthy. When the brain senses a stressful environment, invading bacteria or viruses, for example, a subset of neurons send electrical signals to peripheral cells to get them mobilized to respond to the stress, such as through breaking up tangles, boosting protein production, and mobilizing stored fat. Because electrical signals produce only a short-lived response, the glial cells kick in to send out a long-lasting hormone, so far unidentified, that maintains a long-term anti-stress response. We have been discovering that if we turn on these responses in the brain, they communicate to the periphery to protect the whole organism from the age-onset decline that naturally happens. It rewires their metabolism. It also protects against protein aggregation, said Dr. Andrew Dillon. As a result of the new study, we think that glia are going to be more important than neurons. While the roundworm, C. elegans, is a long way evolutionarily from humans, the fact that glial cells seem to have a similar effect in mice suggests that the same may be true for humans. If so, it may lead to drugs that combat muscle wasting and obesity and perhaps increase a healthy lifespan. If you look at humans with sarcopenia and at older mice and humans, they have protein aggregates in their muscle, Dillon said. If we can find this hormone, perhaps it can keep muscle mass higher in older people. There is a huge opportunity here. In a commentary in this same issue of Science, 2 Stanford University scientists, Jason Wayne Miklas and Anne Brunette, echoed that potential. Understanding how glial cells respond to stress and what neuropeptides they secrete may help identify specific therapeutic interventions to maintain or rebalance these pathways during aging and age-related diseases, they wrote. Dillon studies the seemingly simultaneous deterioration of cells throughout the body as it ages into death. He has shown in worms and mice that hormones and neurotransmitters released by the brain keep this breakdown in check by activating a stress response in the body cells and tuning up their metabolism. The response likely originated to fight infection, with the side effect of keeping tissues healthy and extending lifespan. Why our cells stop responding to these signals as we age is a big question. Over the past decade, he and his colleagues have identified 3 techniques used by the worms to keep their cells healthy and consequently longer lived. Activating the body's heat shock response, for example, protects the cytoplasm of the cell. Stimulating the unfolded protein response protects the cell's energy-producing structure, the mitochondria. The unfolded protein response is the cell's way of making sure proteins assume their proper 3D structure, which is crucial for proper functioning inside the cell. His latest discovery is that glia, as well as neurons, stimulate the unfolded protein response in the endoplasmic reticulum, ER. The ER is a cellular structure that hosts the ribosomes that make proteins. The ER is estimated to be responsible for the folding and maturation of many as 13 million proteins per minute. A lot of the work we've done has uncovered that certain parts of the brain control the aging of the rest of the animal. In organisms from worms to mice and probably humans, Dylan said. Two other interventions also increase lifespan in worms: diet restriction, which may call into play other anti-aging mechanisms, and reducing the production of a hormone called insulin-like growth factor, IGF-1. Dylan's discoveries have already led to new treatments for diseases. He co-founded a company, Mitrobridge Inc., recently acquired as Astellas Pharma Inc., based on the finding that certain proteins help tune up mitochondria. A drug the company developed is now in phase 2 clinical trials for treatment of the damage that occurs when kidneys restart after sudden failure, such as during an operation. He co-founded another company, Proteostatus Therapeutics, to develop a treatment for cystic fibrosis that is based on activating the unfolded protein response to repair ion channels in people with the disease. The discovery about how neurotransmitter and hormones impact the ER could have implications for diseases that involve muscle wasting, such as Huntington's disease and forms of myocystis. Wow. I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.